This is lab test 3 review, and we get started on this one back on page 121 in our lab manual, way back on page 121 in our lab manual with experiment 10 on human genetics. So this lab is specifically about humans, and specifically humans have 23 pairs of chromosomes. So this first question wants to know, where are autosomal characteristics carried? On what chromosomes? What chromosomes are autosomal? Thank you, Alish. 1 through 22. And those are pairs 1 through 22. Forget, you have 23 pairs of chromosomes. Okay, how about sex-linked traits? Where are they carried? The 23rd pair. Good. Now, if you want to move so that it's easier for you to see, feel free to move. If there's another spot available that's easier for you to see, that sounds good to me. Okay, number 2. What chromosomal abnormality is associated with Down syndrome? 1 extra chromosome in what number? 20 what? 21.
What's the 3 for?
You got 3 of them? How many should you get? 2. So you might remember that we also call this a trisomy 21. Down syndrome is trisomy 21. We end up with 3 21st chromosomes. Be able to recognize Down syndrome with a karyotype. In a karyotype. So if you see 3 21st chromosomes, that's Down syndrome. Okay. How can you tell the sex of an individual in a karyotype?
With the Y.
Okay, so if you see 2 big chromosomes at the 23rd pair, 2, they're about the same size and they're pretty big, what do you have? That's a female. But if you see 1 big one and 1 really kind of goofy looking small one, oh, that was wrong. You just see a small one and a big one, what is that? That's a male. Now, listen, guys, Y chromosomes are not shaped like the letter Y. I see people looking at me funny. They're just called Y chromosomes because they're the letter after X. X, Y, Z. You know your alphabet? They're just, but they're small. Okay, so this would be the male. Be the male. Okay. All right, we did some chromosomal abnormalities that were associated with the 23rd pair. Page 125 is where you can find this in a nutshell. Okay, what chromosomal abnormality is associated with Turners? XO, that's correct. How about poly-X? 3 X's. Of course, both of those disorders would give you what sex of an individual? Female. Okay, how about Klinefelter's? XXY. Okay, what would be the sex of that person? Male. And lastly, Jacob's? XYY, and the sex? Male. Make sure you can recognize these disorders with karyotypes. We practiced this on page 125. So you might want to go back and review those karyotypes. Okay, number 5. If a person is homozygous dominant, big A, or they are heterozygous, big A, little a, what characteristic will be shown in their phenotype? Very good, Taylor, the dominant. They're going to appear dominant. So the point is, they could be homozygous or they could be heterozygous. You really can't tell by looking at their phenotype. How about if an individual is homozygous recessive, little a, what trait will show up in the phenotype?
Recessive.
Very good, recessive. Question number 6. You would expect the dominant trait to be the most common in a population. Is this always true? No. Okay, can you give me some reasons why the dominant might not be that common?
It could just get all recessive.
Yeah, but why would recessive be so common? Okay, so it could be natural selection has favored the recessive. That might be a reason. Look at this one. Maybe the founders of that population were all recessive. Then you'd have the founder effect. It's another possibility. Okay, so number 7 says, if I give you information about which trait's dominant, what's recessive, be able to work genetics problems. So I do want to tell you that we did that together, if you will. Page 127. I believe we worked some problems on page 127, bottom of the page, about Nancy and her family. Then look on page 128, there was Joe and Henry. We had cystic fibrosis, Huntington's. Page 129, we did some work on color blindness. And then at the top of page 130, I think we did numbers 3 and 4. So if we worked it, then you need to go back and reread it. So page 127 to 130, go back and re-practice those examples. But anyway, I came up with an example that I thought would be fun for us to just work on, just to make sure that everybody knows how to do these things. And if you'll glance up here, I didn't tell you specifically what is dominant, what's recessive, but I think the way I wrote the question, you can figure this out. Can you figure out, is widow's peak, or a straight hairline dominant? Widow's peak is dominant, and I told you, but not in words. How did I tell you? Capital W. So if you learned the capital letter is dominant, the lowercase is recessive. Okay, we've got to work out the genotypes the best we can for all of these people. So I have 2 people with widow's peaks have 3 children. 2 of the children have widow's peaks, and 1 child has a straight hairline. What are the genotypes for each? Okay, let's start with the 2 people. The 2 people. They both have to be heterozygous. I agree with Alish. Which kid told you they had to be heterozygous? The one that has the straight hairline. Okay, so on the children, let's talk about the children with the widow's peaks first. What do you want to put for the children with the widow's peak? Big W. The second position doesn't matter, and it could be either, correct? So we're going to go like this. What do you think about that? It could be big W, big W. It could also be big W, little w. So we're going to go big W, question mark. Is everybody okay with that? Okay, then there was this other kid that had a straight hairline. What do you want for that kid? Good. But okay on being able to work a problem. And again, I'll tell you what's dominant, what's recessive. Okay. Number 8 is true or false. Sex chromosomes, you know the X and the Y? Sex chromosomes carry only genes that relate to the sexual characteristics or the sexual functions of an individual. That's false. Can you give me an example of something that's on a sex chromosome that has nothing to do with your sexuality? I'll help you. Color blindness. Color blindness is on the X chromosome. Does that have anything to do with your sexuality? No, it doesn't. Hemophilia, being able for your blood to clot, that's on the X chromosome. Does that have anything to do with your sexuality? No, it doesn't. Okay, so there's lots of examples of genes on the X chromosome that have nothing to do with your sexuality. And it's primarily the X chromosome we are speaking of here because the Y has so few genes. Everybody okay on that? Great. So speaking of the X chromosome, traits on the X chromosome are called X-linked. There are some disorders that are recessive found on the X chromosome. And so I have number 9. How are X-linked traits inherited differently in males and in females? So they show up mostly in males. Let's go with that. They show up in males, so males are affected.
And carriers are
And the carriers are females. Good. Everybody's okay on that? Great. Okay, so we're going to have to do some pedigrees. And so I want you guys to look on page 130 and page 131. So if I show you a circle in a pedigree, what does that mean?
it's a female.
That's a female. How about a square?
Male.
Okay. If you link them together, they're mating. How about if you color one of them in? They have the disorder. Whatever you're studying, they have it. Okay, does a pedigree show genotypes or phenotypes? Well, from the phenotypes, you can usually figure out the genotypes. So it initially just shows you phenotypes, but often you can problem solve and figure out the genotypes. Not always, but quite often. Okay, when I say predict how a trait is inherited, page 131, there's your 3 inheritance patterns, everybody. Make sure you review them and the clues so that you know which kind of inheritance pattern each one illustrates. So just for example, I made a little pedigree up here if you want to glance up here. And so before we start working this pedigree, we have to figure out what type of inheritance is going on. Our choices are autosomal dominant, autosomal recessive, or X-linked recessive. I'm going with autosomal recessive also. And first it was recessive because neither of the original parents had it, but one of their 3 kids does. And then I see it showing up in males and in females. Okay, so good. We're just going to use the letter A. So everybody just look up here. What do you think the genotype is for generation 1, individual 1? Big A, little a. Now, which child of theirs told you that? Yeah, generation 2 are their children. It's their 3rd child, but we call that one generation 2, individual 4. What's the genotype for generation 2, individual 4? What kind of A? I know. Okay, this is a male, correct? Okay, look at his mate, the female, generation 2, individual 5. What's her genotype? Big A, little a. That's correct. Very good. Okay, so how about their firstborn daughter, generation 3, individual 4? Big A. How come it couldn't possibly be big A, big A? Because that father, the father is little a, little a. All the father could give its child is a little a. Okay, so everybody's good on pedigrees? Okay, so we're going to have at least 2 pedigrees on the test. And you can't do a pedigree if you don't know the inheritance patterns. So please memorize or learn those inheritance patterns. Okay, so we're going to look at blood type also, because these are great genetics problems. You may remember A is dominant, B is dominant, O is recessive. And I've got Susan is A and Jack is AB. What possible blood types are their children? So I just want to remind you real quickly, this was a table we had in our notes weeks and weeks ago. I just want to remind you, blood type A, you could be AA or AO and be blood type A. And blood type B, you could be BB or BO. But if you're AB, your genotype is AB. And if you're blood type O, because O is recessive, you have to be OO. Now, we're going to work this previous problem. Let me just put it back up there. Susan is the question mark for me. In other words, I want to know, when we work the Punnett square for Susan, are we going to use AA for Susan or AO? We're going to use AO. Why?
We don't know what
We don't know. So you have to assume that she's heterozygous. That way you're covering all your bases. Okay, if you're a genetic counselor, you want to cover all your bases. All right, so let's see here. I've got Susan on the side, Jack across the top here. So we've got Susan and Jack can have a baby blood type A. They can have a baby blood type AB. What would this blood type be? A. And how about this last one? B. So if we had a choose all that apply question, we would color in A, B, and AB. Could Jack have a child blood type O? No. All right. So that's all I have for the first exercise, exercise 10 on human genetics. Everybody okay on that one? Good? Okay. Okay, we're going to go to DNA biology and technology. And I have these three definitions, and they are good. We could do them with words. But I also have this picture. And I remember this from our lecture notes, where each of these arrows stand for one of those words. Each of these arrows stand for one of those words. So let's talk about the first arrow. In the first arrow, it shows DNA being used to make more DNA. Now look at your choices. I got replication, transcription, translation. Which one is this? It's definitely replication. The first arrow where DNA is used to make more DNA is replication. Okay, now we have the second arrow, where DNA is used to make RNA. What is that one? Transcription. Good job. And our last arrow was where RNA is used to make protein. Translation. Everybody okay on the differences in those three processes? Just a quick reminder what the words were referencing. Super. Okay, three parts of a DNA nucleotide. Well, first let me tell you that I think the artwork on page 136 is pretty good. On page 136, I have what are the three parts of a DNA nucleotide? And just real quickly, in the little box that's on the left that's kind of a creamy color, they have two nucleotides there. Hydrogen bonded together? Yep. So what are the parts of a nucleotide? Okay, a phosphate. Did somebody tell me phosphate? 5-carbon sugar. Okay, so I got a phosphate. Oops. A 5-carbon sugar. And a nitrogen-containing base. And on page 136, the bases are G and C in that example. Now, if this is specifically DNA, which this question apparently is, can you tell me the identity of the sugar? Deoxyribose. Okay, class, let me tell you again, sugar will not be an answer on the test. So don't pick sugar because it won't be there to pick. But deoxyribose and ribose will be on the test. You have to know which one's which. Okay? All right, now these bases that I have in the box, and again, we're doing specifically DNA in this question. What could these bases be? A, T, G, and C. Okay. Now, I think you'll agree with me that DNA does look like a ladder, especially the way we built it. Oopsie. Because we took the helix and made it flat, so it truly does look like a ladder. Yes? So if you said DNA looks like a ladder, what parts of the DNA make up the sides of the ladder? That's good. I'm going to use deoxyribose. Why? Because sugar's not on our test, yeah. Do you guys know what rungs of a ladder are?
The steps.
The steps. In case you don't know the word rungs, they're the steps. And so what makes up the steps or rungs of this ladder?
The bases.
The base
Hydrogen.
pairs with hydrogen bonds. That's good. So I'm going to put base pairs.
Well, hydrogen bonds do important.
In a minute they will. I'll go ahead and write it down now. Good question, Ashley. Now, because this was the model that we built, this will be the model that will be on your test. And so, I took a picture of it. It's just this. Okay, and so, let's start with this white thing right here. So let's just call this number 1. What is number 1?
That would be the 5'.
It is the 5' end. And what's the white tube?
The sugar.
No, it's the phosphate. The white tube is the phosphate. And I want to tell you how I know that.
Sugar metal.
Because the sugar is what attaches to the bases, not the phosphate. Bases are always attached to the sugar, not the phosphate. So if you'll check out this picture, see these colored tubes are attached to that black thing, not the white thing. Okay, and then this, because that's the phosphate, Jose's right, this is the 5' end. If that's the 5' end, what's this end on the other side? The 3' end. That's good. Okay, let's come on down to this black thing. It's my second arrow, and I'm going to call it number 2. What do you think number 2 is? Deoxyribose. Good. Okay, now, we don't know exactly what the colored tubes are. It could be C, G, T, or A. But in general, we could say these colored tubes are bases. Are you going to have to know that A goes with T and C goes with G? Yeah. All right, I got one more arrow here. We'll call it number 4. We think number 4 is pointing at hydrogen bonds. That's good. Those are the hydrogen bonds. They're in the middle holding the bases together. Hydrogen bonds. How do you know this is DNA?
Because
It's like
It's double-stranded. Very good. Because it's double-stranded. You have to know that this is DNA because it's double-stranded. So let me show you. So would this be DNA? Everybody look up here. Would this be DNA? No, because it is single-stranded. So instead of DNA, this is RNA. Okay, good. Okay, so number 3 says to predict the complementary base sequence of a piece of DNA. So I thought it'd be fun to actually just practice together. So I put a little piece up here. This goes with number 3, but it's not exactly there. It does also reference table 11.2 where we practiced in our lab manual. But class, as a group, can you go ahead and get me started here? First of all, this says 3', so what would be right here? 5, okay. Go ahead. How'd that go? Everybody feel comfortable with that? No problems? Okay. All right, we need differences in structure and differences in the chemical composition between DNA and RNA. And I'm going to get us started because earlier today you told me that DNA has A, T, C, and G, but RNA doesn't have all of those. One of those is missing. Who's missing from RNA? Thymine, T. So DNA has T, but RNA doesn't, and instead RNA has U. Good. Okay, then earlier today we said that DNA has deoxyribose. So I'm going to squeeze that in, deoxyribose. But RNA doesn't have deoxyribose, it has ribose. And then earlier today you could tell that was DNA and this was RNA because double-stranded and single-stranded. Good. Are you okay on DNA versus RNA? Great. Okay, so what do I have up here now? I took a picture of it. What is it now?
RNA.
This is RNA. And in our model kit, what kind of RNA? Here's your choices, M, T, or R.
Oh, R. M.
What does mRNA stand for? Messenger. This is messenger RNA. So it's single-stranded. Guys, help me out with a few things. We'll just start right here. We'll call this number 1. And the pointer's on those blue pentagons. What is that? Okay, number 2 is on the white tube. Phosphate. And any one of the tubes that are colored, number 3, bases. Okay, I have a real quick question. What would you call 3 of these? A codon. That's good. So here's another codon. 3 bases in the messenger RNA is known as a codon. We'll have that question in just a second. Is everybody okay on this picture of mRNA? Okay. Oh, there it is. What's a codon? What's its function, or what does it code for? Whole proteins?
Amino acids.
Yeah. One codon codes for 1 amino acid, so codes for 1. Can I use AA for amino acid? Is that okay? Abbreviation AA? Okay. Okay, so earlier today when we had our sentence that had DNA with an arrow to make more DNA, you said that was replication. DNA to RNA, you said that was called transcription. RNA to protein, you said that was called translation. So I want to know, what's the name of the organelle associated with translation? Ribosome. Good job. Okay, now number 6 says, if I give you a DNA code, be able to transcribe it. Well, I thought we should practice. So I put up a small piece. First of all, how do you know my little piece up here is DNA? It has T. And then how do you know you're supposed to make RNA from it? Transcribe. So go ahead and get me started. How many codons are up there? 4. Good job. Everybody feel comfortable with that? Good. Okay. I took a picture the day we were doing this, and that day, I remember coming around and checking every group, and I told you that this picture illustrates a process. So the first question would be is, what's the name of this whole process? Translation. This is translation. Okay, let's label some parts. What's part number 1? That's a ribosome. Part number 2. That one's mRNA codons. That's good. And part number 3. That's tRNA. Good. We may have this. It may be true. We'll know on Tuesday. It's okay on this. Great. Now, I zoomed in on just one of these. What did you say this is? This is tRNA. Yeah, so this top thing, this right here, what is that? Anticodon. And how about this bottom thing? Amino acid. That's okay? Right, so the reason I put that picture there is I think it goes with number 7. That picture goes with number 7. What 2 components necessary for translation are carried by tRNA? Well, it's the anticodon. You got to have that to do translation. And you got to have the amino acid. Now everybody knows what an anticodon is, but I'm going to go ahead and put it in words for you. An anticodon is 3 bases. An anticodon is 3 bases on tRNA. Complementary, not the same as, but complementary to a codon. That's why it's called an anticodon. So I thought it'd be fun to practice. Because you guys can do this, but it's been a few weeks. So I thought we'd just refresh your skills here. I put 2 codons up here, and I need you to give me 2 anticodons. Let's do the first one. Go ahead. You're not very good at this. I only heard one person. A-U-A. You're going to have to do anticodons. I'll give you codons, you do anticodons. Okay, I've given you 4 codons here. Now, to do this, next question, you have to have a genetic code.
Well, we need to bring these with us.
No, ma'am, you will not need to bring this with you. It is incorporated in the test. Okay, so we need A, U, G, and just give me the first 3-letter abbreviation.
NBT.
Okay. All right, C, G, C.
ARG.
Arg, good. Is there anybody who's confused on how we're doing this? Okay, great. U, A, U. Tyrosine, good. And U, A, G. So what would happen there? It'll stop. How did you know to use the genetic code and do what you just did? Translate. That's good. Okay, earlier today I asked you to find a sheet of paper that's called electrophoresis. If you were here that day, then you'll be really glad now that we're on question number 9. On the back, we did some notes about our DNA isolation. Maybe we even labeled it DNA isolation. And so what I did is I took some bananas and made a cell extract out of the bananas. Can you tell me why I used the bananas? What role did the bananas play? Good, the source of DNA. Because it was cells. Okay, SDS, it emulsified. This was a detergent, it emulsified. And that means it broke apart the membrane so that the DNA could leave the cell. You had to break up the membrane so the DNA could actually leave the banana cells.
So that's how it's gonna be worded on the.
And then lastly, what was the role that the ethanol played? So it's kind of like a DNA magnet. But okay on that one? Okay, great. So now we're going to go to the front of this same page, and we're going to concentrate on the first sentence at the top of the page. What is gel electrophoresis used for? I heard separate molecules. Upon what basis does it separate molecules? I heard charge and size. So, to separate the molecules had to move. I use the word migrate here, move or migrate. What does it need to make the molecules move? I mean, why do they move? Electricity, good. And I think you'll remember I came by and made sure everybody had electrical flow through their chamber. We look for bubbles to make sure. Okay, still on the front, bottom table, what we did was we looked at a protein's isoelectric point, and we figured out whether the protein would be positive or negative in particular pHs. We practiced that as a class. And then we actually used a pH of 7. Now, I have here, if given a protein's isoelectric point and the pH of the buffer, determine if the protein will be positive or negative. So, again, practice this table. And then also determine the distance it will travel relative to other proteins. So I just drew this up here just to see what you thought about it. Just pretend each color is a different protein. And they all started in the middle, so that's why I put those dashes there. Those dashes represent the wells that would have been in the middle of the gel. And then if you'll notice, I put the positive charge and the negative charge. Okay, so if you just glance up here, which of these proteins are positively charged?
1 and 5 is positive.
Okay, I'll ask again. Which of these proteins are positively charged? It's 1 and 5. Why is it 1 and 5? Because positive charges are attracted to negative. So that's a simple thing you should be able to do. Some of you jump too fast. Okay, so think before you start coloring in your Scantron. Okay? All right, so 1 and 5 are positively charged. That means 2, 3, and 4 are negative. Now, everybody look at 2, 3, and 4. Out of 2, 3, and 4, which one had an isoelectric point closest to the pH given, which I believe is 7?
4.
It is 4. How do you know? It barely moved. Yeah. So if you have an isoelectric point close to the pH, you're not going to have much charge, thus you won't move very far. Okay, so that's what I mean by the second part of number 11. Everybody okay on that? All right, great. It's time to move to the back. Evidences of evolution. Okay, everybody knows evolution. It's the genetic change in a population. And usually over time is included there. We'll talk next week about how fast it could happen. So it could happen, in just a few short generations. Actually, within one generation, you could have a substantial genetic change in a population, or it might be a very slow process. Just varies. That's evolution. Now, evolution, I think you can tell, is a pretty important concept in biology. The class you're taking is a pretty important concept. So I also have this. How does evolution explain the unity of life? Common ancestry. Yeah, that all life has common ancestry. And that's what we've been dwelling on in this class. But some of you are going to also take Biology 2. And in Biology 2, what you concentrate on is diversity. Because there's over 2 million different species on Earth. And so you're going to just take a look at the representative examples of them in that class, Study Diversity. So how does evolution help us understand diversity? I mean, if all life came from one ancestor, why are there so many different kinds of life? There's mutation. What else? What kind of selection? And natural selection has the word nature in it. Are all environments on Earth the same? No. So there are adaptations to different environments. That's very good. So I'm going to put that, adaptations to different environments. That's a huge reason why. Indeed, if the Earth had very few environments, we would not have the diversity of life that we do have on Earth today. But we have thousands of different kinds of environments. All right. Okay, so one day we were studying fossils, and that day we also looked at the geological time scale, which is on page 150. And I don't know, you may have written down what Paleozoic means. Did you? Ancient life. Very good. Paleozoic, ancient life. How about Mesozoic? Cenozoic. Okay. All right, everybody has their fossils. And I want to remind you that the fossils are on Blackboard under course content. You can find a PowerPoint with pictures of the fossils. Everybody's okay on that? What do you have to know about the fossils? Era and common name. Era and common name. Okay, then we started working with a new word. Well, it's kind of a new word. It was a new usage of the word. Homologous, page 154. If you would, turn to page 154 and find homologous up at the top of the page. Same structure, that's right. And we really did utilize those skulls in the back to try and show homology. Like you can look back there and see the bobcat and the domestic cat having high homology. The deer, the goat, the sheep, the cow, they have high homology. Let's see, the raccoon and the ringtail back there, they have high homology. So what does that mean when I say they have high homology? Lots of similarities. Yeah, they have the same basic structure. Okay, good. How about analogous? What does that mean? Same function. I would argue a human hand is analogous to a bird beak. What do humans use their hands for? Object manipulation. What do birds use their beaks for? They manipulate objects with it. Sure they do. It's how they build their nest. They move things around. You sit and watch a bird. It does object manipulation. Does a lot of the same things with its beak that you do with your hands. You can't eat with your hand, I'll agree. Okay? But because their forelimbs are used for flight, they've adapted to using their beak for object manipulation. Kind of cool if you think about it. Okay. Oh, that's a good question. You get to pick between homologous and analogous. Does homologous or analogous represent a closer evolutionary relationship? Definitely homologous. You want to look for similar structure, not similar function. Okay, that same day, we compared on page 156 a human versus a chimpanzee. And specifically, right now, I'd like to talk about from here down. Hey, look, from here down. So not my skull. Don't think about my skull right now, but think about my neck, the trunk of my body, my arms, my legs, my feet, and compare that to a chimpanzee. There's basically one thing that helps us understand the difference between a chimp skeleton and a human skeleton. What is it? The spine is an example of this one reason. I need a reason. Sorry?
Differences in posture?
Posture. Excellent. Our spine is different. Our hips are different. Our knees are different. Our feet are different because of the posture. Mm-hmm. Okay, and then on page 158, we compared the skulls. The skulls. So I have what accounts for the differences between chimp skulls and monkey skulls. So one thing that kind of stands out for me, I'm just going to hold them up again, guys, is the size of this and how round it is compared to that of a chimp. You'll see a big difference there?
Yeah.
Me too. So why is that? Brain size. So that would be the first thing I'd write down is brain size. Brain size. Okay, the second thing I notice is when you look at these skulls in profile, the chimpanzee looks to me like it's got a really big jaw. Yeah, upper jaw, lower jaw compared to us. Look how theirs just sticks out, ours doesn't. Does everybody see that? What would that be due to? Sorry? Diet. Excellent job, diet. So what accounts for the differences between chimp and human skulls? Brain size and diet. Everybody okay on that one? Okay, I just want to remind you, you have to know the skulls in the back. And I do have, again, a PowerPoint on Blackboard that have the skulls. And then on the skulls, you should be able to look at a skull and tell me by its teeth what kind of diet it has. What if you find a skull that has all sharp teeth? It's a carnivore. How about if the teeth in the back are highly ground down? That's an herbivore. What if you find a little bit of grinding and a little bit of sharpness here in the back? Omnivore. That's good. So that's what I mean by what do teeth tell us about an animal's diet. You can tell by looking at their teeth if they're mostly plant eaters, mostly animal eaters, or if they eat a mix. Oh, and on the test I'll use words like carnivore, herbivore, omnivore. Is it okay on that? Great. Okay, comparative embryology is on page 159. I remember one day in lecture I showed you 4 embryos up on the screen and you were supposed to figure out which animal went with each embryo. And frankly, you were very bad at it. You're just poor, very poor. Why was it so hard? They look so similar. Yeah. So I have, how does comparative embryology help determine relationships between organisms? So the more similar embryos are, the more recent their common ancestry. Everybody okay on that one? Super. I like this part. Would you expect a human embryo to be more like a chicken embryo or more like a pig embryo? I agree, pig. And then why? They're both mammals. Good job. Okay, to help you out on this question on biochemical molecules, let's go to page 160. What are some basic biochemical molecules that practically all organisms have in common? Cytochrome C, so protein, there are some proteins that are really similar. What else? RNA, all three kinds. What else? ATP, what else? You're leaving off the obvious one. Thank you. They all use the same genetic code? Yes? Okay. How can you explain that all life has the same basic molecules?
They all derive from the same.
Common ancestor, that's good. They all derive from a common ancestor. Okay, guys, at the very end of that lab, we looked at the antibody-antigen reaction on page 161. Now, this particular day, we used some stuff in dropper bottles to stand for synthetic serum. Let me show you. Today we used these materials. You guys remember this? Yes? And so what we did was we put some rabbit serum in the wells, then we sensitized it with human serum, and then we put serum from 5 different animals into the wells, and we were supposed to figure out who was most closely related to humans and who were least closely related to humans. To figure out who was most closely related, what did we look for? The most white cloudiness or the least white cloudiness? The most. Look for the most white cloudiness. And then the least would be least related. So it's just what you think it would be. If they're closely related, you're going to see a lot of antigen-antibody reaction. If they're not very closely related, you're not going to see much antibody-antigen reaction. Okay, and then lastly, we're going to have this on the test. This is on the back of your fossils. It's the cytochrome C protein clock. I'm going to have that on the test also. So be able to answer these questions or, something like these questions. If you can use this chart successfully, you'll be fine on the protein clock questions. Yes, sir.
The numbers represent millions?
The numbers represent actual differences. So they sequenced the amino acids and then they counted how many differences there were. Good question. Okay, exercise 13, mechanisms of evolution. Class, that was last we met, just 2 days ago. And I'm going to defer to your lecture notes and or page 166. You can see all this on page 166 also. Now, the reason I have a line drawn up there is because the first 2 are just definitions, but the last ones are mechanisms of evolution. Those are actual mechanisms of evolution. But I think you can find all of these things on page 166 if you need to. We've been talking about all these things in lecture. It should all look real familiar to you from Tuesday and from today. So again, the list of words, you want to go back and review their terms and what each one means. Page 166 is a pretty good place to go.
You said the under the line or what?
Mechanisms of evolution. Mutation is a mechanism of evolution. Gene flows, how evolution can occur due to these things. And remember, evolution is the change in the genetic makeup of a population. Okay. Okay, so Hardy Weinberg is coming up. We're going to work a Hardy Weinberg in a moment. But first, what's the purpose of Hardy Weinberg? To know if evolution's occurring or not. So baseline for evolutionary comparisons. To know if evolution has occurred or not. Because evolution is the genetic change of a population. So to know if a population is genetically changing, you have to know what it was so that you can know in the future if it's changed. Okay, now remember Hardy Weinberg said that if a population's not evolving, it's an equilibrium. And that its gene frequencies will not change. So, here's the next part. In the absence of outside evolutionary agents, that means no evolution, how would you expect gene frequencies of a large population to change over time? They won't. Now, when that's happening, we say the population is obeying Hardy Weinberg. It's meeting all the criteria of Hardy Weinberg.
It is an equilibrium or it's not an equilibrium?
It's saying it is. Because the only thing that'll throw the genetics of a population out of equilibrium is evolution. So this is saying no evolution, thus the population is in equilibrium. Number 3, true or false? The frequency of the recessive phenotype, that means how often you find the recessive phenotype, equals the frequency of homozygous recessive genotype. That is true. Because if you show the recessive phenotype, you must be homozygous recessive. Isn't that true? Well, this number 3 is true. Number 4, true or false? Okay. The frequency of the dominant phenotype equals the frequency of homozygous dominant genotype. Okay, I agree. How come that's false? Because you're not accounting for heterozygous. That's good. Okay, I know I've said this before, but let me say it again. You have to know the Hardy Weinberg equation and what each symbol of the Hardy Weinberg equation represents. So page 167 has all the parts of Hardy Weinberg defined for you there. Page 167. It's right at the top. It says where Q is the recessive allele frequency, Q squared is homozygous recessive. P is the dominant allele frequency, P squared is homozygous dominant, and 2PQ is heterozygous. Everybody okay on that? Okay, I promised that to you the other day, and I will deliver. Okay, number 6, given a set of genotypic frequencies, I'm just making this up. Let's just say P is equal to 0.7 and Q is equal to 0.3. Let's just say that. What would you expect the frequencies to be the next generation if the population obeys Hardy Weinberg? The same, 0.7 and 0.3, so I'll write the same. Okay. How about 5 generations later? The same, because it's an equilibrium. It's not changing. How about after 10 generations? Still the same, because it's an equilibrium. It is not changing. If it's obeying Hardy Weinberg, it is not evolving. Okay, so I promised you a Hardy Weinberg, so I came up with this one. Get out your calculators, and they do need a square or square root button. If you have your cell phone, you can use that. But can you use your cell phone next Tuesday? No. Can you use your cell phone on the final exam? No. No. So make sure that you have obtained a calculator with a square and square root button. Okay, I have 400 total fruit flies. 375 have long wings, which are dominant, and 25 have short wings, which are recessive. And we're supposed to do all the Hardy Weinbery. Okay, so last we met, I let you go ahead and work on it, and then we kind of talked about it afterwards, so let's go ahead and work on it, and then we'll see how it goes, okay? And if you are at your table and you have no clue what we are talking about, other people at your table have clues, and they can help you if you're lost. So just let them know they're your buddies. Okay, so let's see what you all came up with. Can you get me started with q squared? You always start with q squared, correct?
Yeah.
So what'd you do? 25 over 400. It's the number of individuals recessive over the total. What'd you get?
6%.
Yeah, 0.06 or 6%, whichever you prefer. Okay, and if you didn't start there, then you didn't get anything else right. Okay, you got to start out correctly if you have any chance at all.
Do you get points for effort?
No, it's multiple choice, so no, you don't. All right, how do we get Q? Square root of 0.06. I don't have a calculator. Can you help me? 0.24?
Yeah.
How do we get P? Okay, I'll need the help of calculator people. I need 0.76 squared. 0.58. Or 58%. Or homozygous dominant. And then I need, let's see, 2 times 0.76 times 0.24. That looks good. Should add up correctly also. Ben?
So just so I know I'm following, P equals 1 minus the square root of Q?
No, minus
No?
It's 1 minus Q.
Okay, so Q is the square root of...
Q is the square root of Q squared.
No, I'm just-
'Cause that's true in math in general. No, I don't mean that rudely. I mean that's just a factoid from math. Okay.
I'm just trying to pick up on it.
Yeah, pick up on it.
Because I can.
So, Ben, if you'll go back and study what each thing stands for, it'll make more sense to you too. So to get started, we took the number of recessive individuals divided by the total because we know what their genotype is. They're little a, little a. And so Q squared is the percent little a, little a. And then to get Q, we took its square root of that, subtracted off of 1 to get P, and then it's just a downhill slide after that. Okay. Okay, I got 2 more questions for you, I think. Is that true? Yeah. Okay, I need to know the difference between the founder effect and bottleneck. Okay, so which one is where you find a population on, we could say, an island, and it's similar to the population on land, but a little bit different? Founder effect, definitely. So I wanted to tell you, because I talked so much about the bay the other day. Remember talking about the ship channel?
Yeah.
Okay, so out in the bay there are islands. Have you ever been out there? And they weren't there before. We made them. When we dug out the Houston Ship Channel, the dirt had to go somewhere. So we just piled it up out there in the bay. So initially there wasn't anything there. Now there's all kinds of plants and animals, so clearly we've had some migrants that made it out there and they colonized it. So the founder effect probably is going on out on those islands just right down the street in the bay. Has anyone ever made it to one of those islands, just on a little boat, gone out in the bay? Did you notice insects there? I got off, well, I don't know if it was just when I went, but we got off a boat. It's been many, many years ago. And darn it, we didn't know. And so we were getting bit up by mosquitoes and flies like no other. Okay, now, in other words, they're doing quite well out there. But if you go in the spring, summer, or fall, you will find all those mosquitoes. But if you go in the winter, you will not, because that large population is now a very small population. What do you call that? Bottleneck. Okay, good. Okay, examples of natural selection pressures. I'm going to get us started. Natural selection pressures could be things like predation. What does predation mean? High amount of predators. In other words, a lot of pressure for being eaten. Yeah, you're going to become prey. Predation is a very strong natural selection pressure. What are some others? Competition, like competition for food, competition for resources, competition for mates.
Changes in climate.
Excellent. The weather, the climate. It's too cold, it's too hot. It floods. Maybe it snows. Could be fire, earthquake, avalanche. I mean, there's just so many possibilities there. Disease, excellent. Those are all excellent, excellent choices. So our last little question is, how does natural selection affect Hardy Weinberg? Does it affect Hardy Weinberg? Yes. It changes it. Yeah, we kept saying that Hardy Weinberg predicts that we would stay the same except if evolution is occurring, and definitely natural selection is part of a mechanism for evolution. Okay, so for our test on Tuesday, I just want to recap. You need a calculator with a square root button, and if you want to do the cut and tape activity, that is due on May 5th at lab time. Okay, great. Oh, and this will be on YouTube if you want to use it while you're studying. It will be on YouTube.